Hello everyone and welcome to today's webinar, Innovating Animal Breeding with Targeted High-Throughput Genotyping Technology. I'm Cassie Soltman of LabRoots and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by LGC Biosearch Technologies. To learn more, visit biosearchtech.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box to the left and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you are having trouble seeing or hearing the presentation. I'd like to now welcome our speaker for today's webinar, Dr. Leandro Neves, Senior Director of Research and Innovation, LGC Biosearch Technologies. For a complete biography on our speaker, please visit the Biography tab on the bottom of your screen. Dr. Neves, you may now begin your presentation.
Okay, thank you for the introduction. Good day, everybody. I hope you all having a good day in where you are. So yeah, today we're going to be talking a little bit about how high-throughput genotyping and some examples how we are applying this to livestock breeding across different industries. So I'll be focusing on the cattle, porcine, and the avian, the chicken industry. And at the end, we can also discuss some potential other species as well. But that's the focus of the presentation for today. And my name is Leandro Neves. I am the Senior Director for Research and Innovation at LGC. And so moving forward a little bit, so I want to start this conversation by saying that we're a biosearch technology. We're a company as part of LGC, and myself come from the Rapid Genomics company. And in essence, we've been genotyping and processing hundreds of projects over the last year. So we have this core competence around agrigenomics. So that's what we mostly specialize and focus on. It's that industrial breeding applications where we have to genotype thousands and thousands of samples. And it's very, very important to note that we come from a scientific background. So I like to say that we are science-driven but commercially focused, meaning that we understand the scientific questions around what we're trying to do, but at the same time, we understand that you have to apply these tools that we're building towards your commercial projects. And that's where the commercially focused mindset comes to help you as you're applying some of this technology that we're going to be discussing today in your breeding program. And the core products that we commercialize as far as Rapid Genomics, we have the FlexSeq, which I'm going to be describing in much great detail today. We also have the CaptureSeq. And lastly, we have whole genome sequencing, which is in essence genotyping the entire content of the genome. And some of the four pillars that we try to bring to every project, to every product that we develop is for these projects and these products to be accurate. So we need to have high quality data. We need to be able to provide you with sufficient information. We also have to have a lot of flexibility because your demands and your questions are always changing. And we need to be able to adjust from our end to provide you the excellent and best service that we can. The products that we built, they all tend to be scalable. So that means that you can do a few samples or you can do hundreds of thousands of samples, and that's still going to work well for that application. And lastly, we know budget is always important. So it's important that we are cost effective as we're presenting these solutions to you. So from an LGC standpoint, we have commercial laboratories in 4 locations throughout the world to better serve customers like yourselves. So we have here on the left side, we have Gainesville in the United States, in the state of Florida. That's actually where I am streaming this webinar to you today. Excuse me. I'm located in Florida here in Gainesville, in the Sunshine State. Also, we have a laboratory in Middleton, Wisconsin, in the United States as well, which I'm showing as the second box on this slide. And third, on the very far right, another laboratory that does NGS genotyping or genotyping using next-generation sequencing is the laboratory that we have in Berlin, Germany. In the UK, we have a laboratory in Hoddesdon who does a lot of DNA extraction and the CASP genotyping that I'm sure many of you are familiar with from a more focused genotyping standpoint. So those are the 4 laboratories that we leverage as we're finding which one is going to best serve you, depending on where your samples are coming from or where it's easier to process your project. The other thing that we have as part of this next generation sequencing solution to you, namely on the FlexSeq franchise, is we give you access through a customer portal to our LIMS, or the Laboratory Information Management System. So that allows you to, in real time, see where the projects are and see the samples being processed. And you can also download some information to help you make decisions, because we understand that we're going to be working with people across time zones and across different countries. So we much prefer to empower you so you can understand where your project is any time of the day. So for example, you might be able to see the overall status, and that's what I'm showing here, where each plate on your project is at that particular time. And you can see in this example that you have plates all over the process. So as we're going through your projects, they're going to be flowing through the lab. You can also go a little bit deeper and look at plate by plate and understand where each plate is. We have some here already completed in green and others still being processed. And you can also view or download information about each one of those plates. And this is available to you as a mobile application as well. So you can download that for either iOS or Android phones. If we go a little bit more into what exactly are we doing as LGC to enable your breeding program in livestock. So you can see, typically, I think of this as a pyramid where we have whole genome sequencing at the bottom, where we're generating a lot of markers. So this pyramid is really reflecting how many markers we're going to be genotyping on your population. So whole genome sequencing, whether you're going to be doing this at a low pass or a high depth, it's going to provide you hundreds of thousands, if not millions, of loci that are going to be genotyped. And if we go a layer deeper, so we genotype some fewer markers, but still quite a bit, we can provide you with that CaptureSeq application. And then going more focused, we have the FlexSeq, which usually goes from 1 to about 45,000 targets. And lastly, we have the CASP, which is a PCR-based technology that allows you to go from 1 to, let's say, about 200 markers or so. The interesting bit is, when do you apply each one of these in your program? It's a question that you would the best person to answer. But from what I see it, we might have on this high density of markers, we're going to be more when we're more in that pre-breeding marker discovery or germplasm characterization. So that's when we're really trying to explore and do more research on your animals and the samples that you're genotyping. So a lot of times people leverage this to do genome-wide association studies and then find candidates that are very interesting from a genetic standpoint that they can then use to do selection on. And when we go on a smaller number of markers, such as the FlexSeq or CASP, that's usually what we think about industrial genotyping, is when you're going to be doing high throughput genotyping. And typically that's done for either parentage identification or genomic prediction, genomic selection, when you're actually using that genome-wide information to predict the genetic rank of the individuals that you're trying to breed. And typically on this, the FlexSeq and CASP side, this is available to you either as a service, which I'm going to be discussing more today, but also on a selected basis, we work with you on a kit as well to enable your laboratory to process samples. So that's the way the pyramid goes. But as you can imagine, the number of samples tends to increase the less markers you're genotyping. So that's why we see a lot of the demand, a lot of that high throughput genotyping comes more on the FlexSeq and the CASP side, because that's when you have a lot of individuals, a lot of animals that you want to genotype, but with a selected number of markers here. The question that's important asking, though, is, why are we doing this? What do we gain by applying molecular markers or applying genomics in our breeding programs, right? And I want to pick one example that I was able to find from the company PIC, or Porcine Improvement Company. And that's the example that very much exemplifies this. So when you see this this result, it's very interesting to notice that on the x-axis we have the years, and on the y-axis we have what's being reported as the genetic index, or the gain that they have on the index value, right? That represents the genetic merit of an individual, and how that's changing over years. So it tends to be growing. It was growing in a very healthy and sustainable pace at the PIC company. But you can see that at one point it changed in inflection, and it starts to be a little bit more steeper. The gains are large, right? And that's when they started to implement the block on their selection using molecular markers to be able to identify the best individuals, right? And so if we think about on this particular example, that's that you can see the reference to that example here, had they been doing what they were doing before, I would take the liberty to say that that's probably the trajectory that breeding program was going to take. It was going to continue to grow, but at a certain rate that was somewhat fixed. But by adopting molecular information, adopting genomic selection, you can see that they've increased the slope quite a bit and are able to achieve higher genetic gains at a faster pace. So to me, that's a great example to show how animal geneticists are applying this kind of technology in their breeding program to leverage better the germplasm that they have and convert that into higher genetic gains for the producers worldwide. So now we're going to go a little bit deeper into the FlexSeq technology, and that's what I want to explain to you, how we are enabling some of these companies to adopt genomics in their programs at a high throughput scale. So FlexSeq, it's a technology that we developed and we have we've been commercializing for a few years already in the livestock environment. So I want to show you some of the examples that I have for different species that we've been adopting this on. And I think a common thing that you're going to see throughout this presentation is that we typically start with a larger panel. So for example, I'm showing here what would be a FlexSeq high density genotyping panel. And typically, density depends a lot on who is asking, but let's say that the high density would be from anywhere between 20,000 and 30,000 targets on the genome that we're characterizing. And that's where we begin. We begin with a higher density that allows you to really sample well that diversity that you have. But we can also provide you another tool, which is contained within the larger set, that is a mid-density genotyping solution. That, let's say, that's going to be between 7,000 and 10,000 markers of the genome that, again, are contained within the 20 to 30,000 high density that you have. But a lot of times that's still more markers than what you need, and you might want to have an even more focused application and perhaps impute your data set to a larger number as you're genotyping a higher number of individuals. And that's when we come and provide you a FlexSeq low density application that typically ranges from 1 to about 5,000 targets of the genome. So this Russian doll approach, if you will, allows you to really choose what's going to be the best approach for your population at a given time. So you might have a subset of your samples that you want to do with a 20,000. You might have a target. You might have another subset of your population that you want to do with a lower density, perhaps a 5, 3, or 7,000. So that's the idea. When we allow you to have this concordant data set that provides you with a tunable genomics, so you can choose what markers are best for each application. And I'm going to be showing this throughout the presentation for these 3 species here: pigs, chicken, and cattle. But before we go into the examples, I want to take a few minutes to explain what is FlexSeq. How are we actually doing this genotyping in this quite challenging genomes? So before anything, we have to design the probes, because FlexSeq is a targeted genotyping, which means that we're going to be going after specific regions of the genome. In the case of this species that I've showed you, we have readily available panels that we've built, which we call the industry standard series, because they've come from this accumulation of data that the industry has, and we try to create one panel that would work for most people in most applications. But you can see on this diagram that I'm showing here 2 targets, but this could be, like we discussed, 5,000, 10,000 targets. But on these two targets that I'm showing, you have one SNP that is represented by the allele C or the allele G, and you have another SNP that's represented by the allele A or the allele C. And for each one of those, we design a left probe and a right probe. And in fact, these probes are double-stranded, so they tend to bind to both strands of the DNA, which brings some advantages to the application and the data. So we're going to be designing these double-stranded probes. They're going to be hybridizing to the adjacencies, so to the left side and the right side of your target. And the next step that we do is to complement the region in between the probes. So we are going to be sequencing the region that's spanning between those two probes, which contains the target of interest, but it might also contain other variants. So you can use FlexSeq as a discovery tool to identify markers that are segregating near your target, your marker of interest. But once we complemented that space in between the probes, we have the molecules that I'm showing on this lower part of the diagram. And once we have these molecules, we go through another step, which is to go do a PCR where we're going to be indexing. So the PCR is using universal primers. It's just really indexing and incorporating those molecular identifiers for each sample, so we can combine all these samples into a run on a sequencing machine, which I'm showing on step 3. We take all those molecules from different samples, combine them, but because each sample had a specific molecular index, we're able to deconvolute that data when we receive it and separate that data between samples and also between targets, because we know what regions of the genome we're trying to sequence. And once we gather that sequencing data, the next step is going to be to process this into useful information for use. So we're going to be doing the bioinformatics to call the SNPs and provide you with, in essence, a spreadsheet that allows you to know for each individual what's the genotype at a given site. So that's what FlexSeq is doing. It's a targeted genotyping that allows you to focus on the specific regions that you want to genotype. And it's a very fast and high throughput application. And to discuss a little bit how this compares and contrasts with other technologies that you might have seen in the market, I want to present some of the advantages of FlexSeq here. So the first one that I've mentioned a few times is that it's a very high throughput genotyping. So we call this ultra high throughput genotyping because it was designed from the ground up to be applied in a routine large number of samples application. So it was designed to be simple. It was designed to be effective. It's a sequencing-based genotyping, so you can scale the number of samples quite easily without having to do a lot of investments from a capital standpoint. At the same time, it's a very simple reaction in the lab because we use one plate that we keep adding reagents to that DNA and making the reactions happen on the same plate. It simplifies waste. It reduces waste. And simplify the application in the lab as well. And it's targeted genotyping, like we discussed. So it concentrates the resources where you wanted them to be concentrated. So if you want to sequence 10,000 targets, you're going to be sequencing 10,000 targets, and you're going to be genotyping about 10,000 targets. If you need more or less, we can adjust that as well. And very important for our customers that this allows you to bring legacy data. So if you're already genotyping, like many of our customers are, with another technology and you want to adopt FlexSeq, we can still target the vast majority of the targets that you are currently genotyping. And we can also do more if it comes time for you to perhaps change that panel. That's something that we can do with you as well. The other component is that it's a very flexible application, I guess, hence the name, right? But it is suitable for any species, for any germplasm. So we don't have restrictions as far as the SNPs having to be SNP-free around them, so we can design probes. Now, these probes, they tolerate diversity. They can be quite effective even in complex germplasm. You can adjust the panel over time a lot easier than you can do with other technologies. So, for example, if you're going through your program, being genotyping with us for 6 months or 1 year, and you realize that you find through your research that there is another target or a few targets that you would like to incorporate, we can incorporate those targets into the genotyping panel very fast without significant costs and without disrupting your genotyping routine. So very quickly you can adjust to your R&D program that's generating new targets for you there. So that's the idea, right? You can adjust the panel over time, and you can also adjust the marker density that you need. And then lastly, I want to highlight that FlexSeq is a hybridization-based approach. And you might see some alternative methods that are based on multiplex PCR. Typically, what that means is that you have more uniform data sets than multiplex PCR, because we are doing one hybridization, and that hybridization is going to be allowing the probes to find the right targets. Versus if you're doing, for example, a PCR, every cycle there is a chance for that primer to bind in a wrong location of the genome or create some level of allele bias. And if once that's created, it gets propagated more and more through the cycles as you do 20, 30, 40 cycles, depending on the application. But if you're doing one hybridization, that's a more effective and a cleaner way to genotype those samples that leads to more specific and on-target data sets, like we're going to discuss here in a little bit. And it also allows us to have this high density if we need to. We can genotype up to 40, 50,000 regions of the genome. So that's some of the advantages. And I hope by showing you these 4 examples, you can see how FlexSeq is accurate, flexible, provides this scalability, and at the same time cost-effective. So how do you begin applying FlexSeq in your program? We like to say that you got to follow 3 easy steps. So the first one is to design or identify a panel that we already have readily available for you. So this could be for the species that we're going to be discussing on this webinar, but it could be for others as well, whether maybe you want to do genotyping on turkey or sheep and other species that we don't necessarily have an ideal panel for you. We can make that panel for you. So that's the first step. We're going to be having some discussions with you, understand your goals, and how we can support you. And then the next step is going to be to validate that panel that we're going to design for you. So you would typically submit some samples for us, and we would genotype 96 to 34 samples, so we can provide you with some data of that panel, and that allows you to see how that panel is behaving and whether it needs to be made any adjustments or fine-tuning, for example. And then lastly, once you have that panel validated and designed and validated, now you can begin your routine genotyping. So your samples will start to flow through the lab, and they'll be delivered on a fast turnaround time for you. And you can apply that information for different goals, such as genomic selection or parentage, depending on what your goal is. So the next block of our presentation here, of our webinar, I want to go through some of the applications that we have, particularly in livestock breeding, which I know is the main focus for the audience here. So if we focus first, just give me 1 second for the slides. Okay. So if we focus first on the swine breeding side of the business that we have, we've been processing a lot of samples for different customers, and I want to give you a flavor or an indication of how we've been doing this for some of our customers. So the first one is really, I want to highlight this porcine 3K panel, which we call P3K. And that targets about 3,000, a little over 3,000 markers that we've selected from industry available and proven data sets. And these are known to generate good breeding value and good breeding evaluations across a range of germplasm background there. So we've developed that panel, and it's optimized and readily available. And when you think about the performance of that panel, it behaves quite well. So I want to show you some of these metrics that we typically use, and you're going to see that being a common thing throughout the presentation. So let's take a little bit of time to go through these metrics here. So the first one is, how does it agree with other technologies, like whether that's going to be, say, an array-based technology or other sequencing technologies such as whole genome sequencing. And we tend to get 97, 98% agreement between these technologies. So it's a very high agreement. And then I suspect that some of those were probably right from our genotyping side compared to alternative methods, because we're using sequencing as a proxy, and it's a bit less biased than potentially other technologies can be. So, but regardless, it's a very high agreement with other technologies. And what's very important is to see how concordant our data set is from one genotype to the other. So if you were to genotype technical replicates, so let's say if you're genotyping the same sample twice, what is that agreement on the genotype calls? And we see about 99% agreement between those genotype calls, which is what you want. You want your data to be the same day in, day out, whether that's going to be in the beginning of the week or at the end of the week, beginning of a month, or during the summer or winter, right? You want to have that same consistent genotype. And that's what we've been demonstrating over time. And then lastly, once you build a panel, you want that panel to deliver. You want that panel to give you what you need. So the other metric that we measure a lot is the marker recovery. So you design a 3,000 panel. How many markers do you actually get when you're genotyping this day in, day out? And we tend to be above 95% for an optimized panel like this one. In fact, you can see on the right side of this slide where I put some of the graphs, and you can see on the dark blue that we were actually getting about 98% for this particular panel. And the last one that I want to show in this slide is going to be the on-target sequencing. So on-target sequencing means that out of the reads that you've obtained from the sequencer, what percentage of it goes to the target that you're actually trying to genotype? And that means that the reaction is very efficient, right? You're not generating off targets, or you're not generating reads that cannot be genotyped. And for FlexSeq, we tend to see over 95% on target. For this panel specifically, you can see on the teal color on the graph that we have about 98%. And the last metric that we use a lot is the uniformity. And this particular panel behaves very well, but I'm going to show you this in another slide here. That is the uniformity of this panel. And we usually plot this as a log plot. So on the x-axis, we have the markers. So we're going to have from, let's say, the first marker all the way to the last one, ranked by how much data they've been receiving from the sequencing side. So you can see that we got about 3,000 targets for this particular panel, as we discussed before. And on the y-axis, we have the amount of data that was allocated for each one of these targets, converted to a log scale. What we want to see is a very horizontal graph. We want this graph to be as flat as possible, meaning that every single target on your panel receives the same amount of data. You do not want to see too many of those targets getting too many reads, or too many of those targets not getting enough reads. So that uniformity is what I'm going to be presenting. And people have different metrics to provide the uniformity. On our lab and our team, we like seeing these log plots because they're very transparent as far as how the data behaves. So that's the example that I want to show you in porcine. This panel is readily available, and you can utilize that on your breeding program if you're interested. The next one, I want to shift gears and show you the story of how we've been building solutions for cattle breeding. And we started with the cattle breeding from a very comprehensive approach. Okay, let's see what the market's using and what kind of markers are mostly interest people most based on their breeds and their application. So we began with this industry standard series for cattle, and we've developed a starting panel that has about 46,000 targets. And that, again, was developed from a set of industry-proven data sets where people are using these for selection or for marker-assisted prediction of key phenotypes as well. One thing that this panel has is that we've collected from the industry a set of targets that are very important for detecting some genetic abnormalities or conditions that you want to screen in your population. I'm showing some of them here that they're segregating a lot, or very important, I should say, in the Angus breed, or Hereford, Simmental, Brangus, and Wagyu as well. So you can see that there are different ones here: Horn-Polled, Deluder, and a few others. They're important for that team, and they want to know what the genotype for those animals are. So we try to capture those targets. And lastly, we also included markers that come from the population, the publication with Mari, Sachi, and Garrick, where they describe a set of markers that's commonly known as MSRP. So we're building our panels to contain a large number of those markers that are important to the cattle industry. So how does this 46K panel behave, which we understand is a very large panel when you're thinking about targeted genotyping, but the panel still behaves amazingly well. So we were able to recover over 90% of those, 80% of those targets. And the data on target is almost 90%. So we still get quite efficient sequencing of those of most of those targets. And just to show you the uniformity for this plot, for this panel, you can see that the plot's still very homogeneous despite the large number of markers. So this is a great screening panel if people just want to understand the population. But what we've done subsequently is to select the best markers. So we chose some of the in the middle of the distribution and very important key markers as well for different groups. And we've built our HD panel that we mostly commercialize, which is a 24K panel. And that panel is optimized again to target industry-proven loci, those genetic abnormalities and conditions, and contains a lot of the MSRP markers. And I wanted to show you two examples of how this panel behaves. So you can see on the left graph our familiar now uniformity plot, where you can see several individuals plotted on that, the data for several individuals plotted on that plot with different colors. And on the x-axis, you can see the regions that we're targeting. So again, we got about 24,000 for this particular panel. And on the y-axis, you see how much data got allocated to each one of those targets. And we're targeting those 24,000 regions. And on average, we're consistently getting above 20,000 of those 24,000 as we're genotyping a broad set of individuals. So we usually tend to deliver 20,000 targets, as I'm showing on the right distribution here, where you can see the box plot hovering a little above 20 to 21,000 targets. So it delivers high quality data when you want to screen a large number of regions on your genome. And as we discussed, a lot of times you might want to do fewer than those 24,000. So we're also offering you a mid-density solution in the 7,000 loci range that captures some of those 24,000 targets. And this panel behaves very well as well. If you look on the two graphs, on the left graph, you can see the distribution. So those 7,000 markers, you can see a very horizontal distribution, meaning that we're recovering most of them within a certain deviation from each other. And on the right side, the box plot is showing how we are recovering in the range of 7,000 to maybe 7,100 across a large number of individuals here. So it's very consistently genotyping the same targets. So that's important to realize, because sequencing can and is delivering reproducible and repeatable data sets to your breeding program. And that is very important to highlight here. So in a nutshell, in the case of cattle, we have whole genome sequencing. So for some applications, we usually begin with genotyping throughout the entire genome and sequencing maybe a few hundred to a few thousand samples on a breeding population per year via whole genome sequencing. But we also offer on a larger number of samples. Typically what I see people doing is something in the range of a few dozen thousand to maybe 100,000, few 100,000 samples that they're going to be doing on different densities. So they might be doing on a 24K on our mid to high density, or they can be using a mid to low density there on the 7K. So that's the solution that we have for cattle. And the next step, we have a solution for chicken as well. So I want to show you some of what we've been doing in that front. Very, very similar thing to what we've been discussing so far. We begin by identifying a higher density panel that works for that population and for that species. and then we do a lower density that's contained within the higher density. So I'm going to be showing you the examples here. We first began with about 16,000 targets that we have for chicken. These are going to be sequencing some of the microchromosomes. They're going to be sequencing the sex chromosomes and also throughout the genome as well. And we've selected that to provide a low density genotyping for the industry that hovers at about 3,000 targets. And how do these panels behave? So we get familiar to this uniformity plot. You can see that we're targeting here about 3,500, and this panel behaves very uniformly across the range of the markers that we're trying to sequence. And the missing data is also very low. So when I ask on the left, looking here at each one of those x-axis, on the x-axis, each one of those box plots represent a plate that contains 96 samples. So you can see that we have seven plates to be precise on this panel. And we on the x-axis on the graph on the left, I have the percent of the loci that have less than 10 reads, which we think they're not very confident. So we consider them as being percent missing if they have less than 10x sequencing depth. And you can see that it's hovering that very few percentage point, maybe 3% of the data there, 3 to 5%. And the vast majority of the samples are behaving well. And on the right side, we have the recovery of the loci. So we are recovering consistently above 3,000, about 3,200 targets for this panel here. And one question that it's important highlighting is what is the agreement of the data when you're doing some individuals with higher density, some individuals with lower density? We already showed that the same panel, when sequenced same animal twice, is going to be very concordant at a 99% rate. But what happens when you do different panels, high density and a low density? So I'm showing here some individuals that we've sequenced with the HD panel and the LD panel. And the genotyping concordance is also very high, despite being a different synthesis of oligos and a different hybridization condition. But the markers that are present across both panels are very concordant. So that allows you for a very strong imputation of your data when you're going from lower density to higher density across your population, which gives you a very powerful tool to apply in your breeding population. So just to show here on the y-axis, we have the percent concordance between biological reps, because these were different samples that we've genotyped with the same panel, and we've asked what is the genotype concordance on those samples, and we can see here hovering about 99% between the two panels across 96 samples that were genotyped on both panels. So that's the common thing that I wanted to explain to you. We have these again, this Russian doll approach where we have whole genome sequencing if you have to generate a lot of data, and then you can choose from a higher density, a medium density, or a low density panel on FlexSeq, and apply this with high concordant data sets, high concordant data to your breeding population. And in summary, I hope I showed you that we optimized FlexSeq to a variety of livestock breeding programs, and we can help you do the same for your breeding program or for your species. And FlexSeq has shown to be very adaptable, regardless of the species and the goal that you have. It's a very accurate and high concordant genotyping tool that is done via next generation sequencing. It's resource efficient because we are optimizing the amount of, or minimizing the amount of plastics that we're using, and optimizing the amount of sequencing that's required as well to be very efficient for your to deliver data sets. And one of the reasons how we can do that is by doing targeted genotyping. So we can sequence the exact regions that you need as you grow your breeding and develop your breeding programs. The FlexSeq is, in our mindsets, very industrial. So we want to be able to deliver data to you on an industrial scale At the same time, never forget our scientific expertise and our roots that allow you to understand what you're trying to achieve and how we can support your breeding application there. And then lastly, we focus on adding value to customers like yourselves. And I'd love to have conversations about how we can do that and have you join the family and work with us as well. So with that, I'll take any questions and actually happy to have some discussions as well. So thank you very much. And for those of you who joined me until now, have a great day.
Thank you, Dr. Neves, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just type the question in the Ask a Question box and click Submit. We'll answer as many of your questions as we have time for. Let's go ahead and get started. Our first question here is, how does FlexSec technology differ from other genotyping methods in terms of its scalability and flexibility?
Yeah, no, that's good. So if we compare with, for example, whole genome sequencing, the advantage here is that we can put a lot more samples in a lane because we're doing targeted genotyping. So we can actually focus the genotyping on regions that we want to sequence, like we discussed. And that scales a lot better as well. If we compare, for example, with, say, microarrays or SNP chip arrays, then there are several advantages of the FlexSeq technology. From a scalability standpoint, it's a lot easier to do more samples. So if you have to process thousands and thousands of samples, it will be a lot easier to do with FlexSeq compared to, say, an array, just because you can, the lab components are easier, and also you can bundle, you can pull more samples in one sequencing run. The flexibility, it's a similar answer. We can adjust the panels a lot quicker with FlexSeq. So if we have, for example, to incorporate new targets that you develop through your R&D, we can very quickly incorporate that on FlexSeq compared to, say, a SNP array. And so that gives you some of the pros and cons relatively to scalability to FlexSeq.
Wonderful, thank you. Our next question here, how can you apply this technology to lab species such as mice and rats or other larger animals used in research?
Yeah, it would be very simple and similar to what we've done for other species. So we're going to start asking you to provide us a list of targets. So some customers in some of these model species like mice and rats, they might have regions that, for example, they've added it, so they have added targets, or they might just have some regions that they want to genotype. So you would provide those sequences, those regions to us. Typically, they can be provided as a BED file, BED. So these are just coordinates on the genome where you would like us to sequence, to focus our sequencing. So you provide us the genome and the BED coordinates, and that should be enough for us to begin designing the panel and provide you the genotyping tool that we need for that species, whether it's going to be mice, rats, or any other species for that matter. And the number of targets can go, like we discussed during the presentation, they can go from a few, few dozen or a few hundred to several thousand to dozens of thousands of targets. So it's very tunable to your specific needs. But thank you.
Wonderful, great questions coming in here. Our next one is, sample prep is always important. What do you use for various sample types?
Yes, it's very important to control for sample input because it helps a lot if you start with the right quality. From animals, a lot of times we see people collecting tissue bunches. So, for example, in the case of cattle and porcine samples, they will usually be ear bunches that will be collected in a buffer, in a stabilizing buffer. And there's some manufacturers that produce these collection devices. So those are the ones that we typically use for cattle and porcine. We've also used blood cards. We've done as well semen. Those are a little bit more challenging. They require more work to get it done right. From an avian standpoint, chicken standpoint, blood is always a great source of DNA, being nucleated, animals with nucleated red cells. So that's usually what we tend to process from an avian standpoint. But yes, it's important that we collect them right, we store them right, and we in advance prepare for them in the lab.
Great, thank you. Our next question here that we have is, are there any limitations or challenges associated with importation of Flexi technology in livestock breeding?
Yeah, not really. We are very used to working with customers from all over the world. We've done projects with so many countries by now. We just have to plan in advance. So the first step will be to work with you to understand how many samples you're going to be sending, what type of samples you're going to be sending, and then we will apply for the right permits with the government agencies to be able to receive those samples from your country to, say, the United States, where we have one of the laboratories, and like I mentioned, I'm located in Gainesville, Florida, so that will be one of the labs that will process the samples. But we just have to work with the right government agencies and with you to understand what we need to do to import those samples. Conversely, if you'd like to run these samples in your laboratory, so we would have to send you the reagents and the kits for you to run in your laboratory. That's a similar story. We're going to have to understand what are the limitations and importations that we have to go through, but it's doable.
Great, thank you. Our next question here that we have is, do you have any genotyping solution for buffalo and Bos indicus, specifically buffalo and cattle breeds from Pakistan?
Yeah, well, the buffalo, I'll invite you to be our first customer. We actually we don't have buffalo panels yet, so please send us a message. Hope to work with you and create another species. The From a Bos indicus standpoint, I would suppose that the cattle panels that we have will work to a degree on the Bos indicus. So we just have to understand exactly how much we're going to have to tune to be able to make them a little bit better for your specific population, but they might be a good place to begin.
Great, thank you. Another question here is, shall we able to exhibit the technology or in research phase?
So if I understand the question, the FlexSeq Technologies is ready to be commercialized. It's available. We are producing and manufacturing and selling this over a few years already. So it's a mature technology that we can offer you. So it's not really, you can apply that in a research phase, but you can also apply this through your routine breeding program. It's something that we are very comfortable offering for you.
Thank you. We have a couple more questions here. If you have questions for our speaker, again, just a reminder, submit them into the Ask a Question box in the lower left corner, and we'll get to as many as we can. We do have a few minutes left here in this webinar, so we'll go ahead and move along. Our next question here is Flexi currently being used in any aquatic breeding programs?
Yes, we have some initial discussions around aquatic breeding programs. Technically, it's totally doable, and we're beginning to have those projects, but they're not as mature as we have for the other livestock species that I've presented. But absolutely, we can definitely work with you from an aquatic standpoint, develop the panels for the specific species that you need, and go from there. But so there's no problems with that. We just need to understand exactly what the species is and how we develop those panels for you. But yes, please go ahead. Let's go ahead.
Wonderful. Another question here. What are the advantages of targeted genotyping by sequencing?
Yes. So one of the major advantages of genotyping by sequencing in a targeted standpoint is that you can control the regions that you want to sequence. And that control allows you to choose how many regions you're going to target. It also allows you to choose what regions you're going to target. So you can avoid complex regions, for example, that tend to be present when you're doing a genotyping by sequencing that's not targeted. So you can avoid those regions that are more complex, and you can focus on the regions of interest. And that's very important in livestock, for example, where we have information about some regions of the genome they're very important to certain traits. They're key to certain traits, and you want to make sure that you're genotyping them with high accuracy and high quality data. So these would be, for example, the conditions, genetic conditions and abnormalities that we've discussed. So it's very important that we know how we're genotyping them with accuracy. So you can use that information to make your breeding decisions. So those are some of the advantages of targeted genotyping by sequencing, is that flexibility that it brings you and the control that you have over the regions that you're going to genotype.
Okay, wonderful. And we have another one in here. What number of samples do you suggest for an avian species when doing genotyping?
If it depends on what you're going to be applying that genotyping for. But let's first answer, if you're just interested in validating FlexSeq, so you just want to know how well FlexSeq works for your specific population, typically you can do that with 96, no more than 384 samples. So that with that number, you can already have a very good validation of that technology in your population. But now if you want to apply that genotyping towards certain applications, so let's say if you're going to be doing genomic selection, you want to have as many samples as you can, but typically this will be in a few thousand at least to begin with. So you can develop your models and then start applying your prediction models on your population. But that's very much population specific, right? If you're going to be doing, for example, some more limited analysis such as QTL analysis, you might be in a few hundred to maybe in the low thousands. So you can have that representation of the linked equilibrium blocks. And lastly, if you're going to be doing GWAS, which it is possible to do with FlexSeq data, you want to have, if your population is not very structured, so it's more unstructured, so you have a lot of LD blocks. So you want to be having at least a few hundred, maybe 500 samples to begin having some statistical power. But again, that's very general rule, and we need to understand more about your population and your breeding objectives before we can describe this a bit better.
Okay, wonderful. And it looks like that was the last question that we had here for our Q&A session. Did you have any final comments for our audience, Dr. Neves?
Oh, let me see.
It looks like one just came in.
Yeah, one just came in here.
What is the innovation in buffalo breeding?
Yes, I'm not very familiar with buffalo breeding, to be honest. I suspect that it's going to be similar to what's done for cattle. The genotyping would be very similar as well, so we can design a panel that is applied to buffalo breeding and that works for your population. So you would have to provide us with the genome. We can even identify with you some of the targets that we want to characterize, how many targets and things like that. And that would allow you to start applying this in the buffalo breeding program that you have.
Great, thank you. Okay.
But as final remarks, I really thank you for watching the webinar. I hope you've enjoyed it. I know we could not cover everything, like there were some good questions at the end, but at any time, just feel free to reach out to us. You will have your contact as well, so we can reach out to you. But I look forward to working with you on so many interesting projects that we've done over the years, and I hope that yours will be the next one. So thank you for watching, and you have a good day, whatever part of the world you are.
Thank you again, Dr. Neves, for your time today and your important research. We would also like to thank LabRoots and our sponsor, LGC Biosearch Technologies, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Any questions that we didn't have time for or those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage that you share that email with your colleagues who may have missed today's live event. And until next time, thank you and goodbye.